2024, revisiting 3 old papers, starting with title Extended Y Chromosome Haplotypes, resolve multiple and unique lineages of the Jewish priesthood. Michael Hammer, in his 2009 paper titled Extended Y Chromosome Haplotypes, resolve multiple and unique lineages of the Jewish priesthood, highlighted that chromosomes carrying the original Cohen modal haplotype, CMH, are not exclusive to Jews but extend to various Near Eastern populations. However, the extended CMH, a more specific marker, indeed showed significant prevalence among Jewish populations, particularly the Kohanim. Published online 2009, Ayug 8. Michael F. Hammer, Doron M. Behar, Tatiana M. Karafet, and Fernando L. Mendez. People, please remember Mendez because he is the man behind our next paper published only 2 years later, titled Increased Resolution of Y Chromosome Haplogroup T defines relationships among populations of the Near East, Europe, and Africa. Authors, Fernando L. Mendez, Tatiana M. Karafet, and Michael F. Hammer. Source, Human Biology, 2011. Published by Wayne State University Press. Our third source paper is Portuguese Crypto-Jews, The Genetic Heritage of a Complex History by the Institute of Molecular Pathology and Immunology of the University of Porto, Portugal, 2015. 6 years after our first source and 4 years after our second source. The main premise of the 2009 paper was establishing that specific haplogroup was over 5 to 8% total of the data that was available at that time. J is overrepresented after family tree DNA sponsored the overwhelming majority of the sponsored Big Y 700 kits for haplogroup J, giving J an unfair advantage. There are credible claims that Haile Selassie as well as Rambam are haplogroup T. Those samples' results at first were revealed publicly, but then they were made anonymous, placed on non-disclosure due to the iconic nature of these individuals' status. Haile Selassie has 1 million followers who believe he is king of king-returning Lion of Judah and God. Rambam is considered the greatest decisor ever. All of the communities in the land of Israel and the Arabistan and the Maghreb practice according to his word, and accepted him as their rabbi. These are men of great stature and even worship in the case of Selassie goes. It makes sense that because of all the work being done on the CMH, they would hide these results and leave T out of the 2009 paper. In the supplementary information, many samples weren't used, and we find Jewish Ts in Syria, Argentina, and Poland. The supplementary information appears to be cherry-picking and intentionally leave out the results from the different population from the Jewish world. I argue when they use a population, for example, out 83 Moroccan Jews and 74 Yemeni Jews, they only show 8 of each in these groups. The hybridization of these 3 papers and the recent data. Michael Hammer is a prominent geneticist based at the University of Arizona, recognized for his pioneering work in the field of human genetics, particularly in exploring the ancient lineages and genetic heritage of diverse populations. One of the significant discoveries attributed to Hammer involves the Y chromosome analysis that linked Thomas Jefferson's male lineage to a possible Sephardic Jewish ancestry. In his investigative work, Hammer compared the Y chromosome of Thomas Jefferson with those in his extensive database of Y chromosomes. He found close matches with 4 other individuals, including a perfect match to the Y chromosome of a Moroccan Jew. Additional matches were discovered with another Moroccan Jew, a Kurdish Jew, and an Egyptian, differing by only 2 mutations. Hammer posited that these genetic similarities hinted at the possibility of Sephardic Jewish ancestry for Jefferson, although he emphasized the tentative nature of this interpretation. Sephardic Jews are descendants of Jews who were expelled from Spain and Portugal after 1492 and settled in various parts of Europe, North Africa, and the Middle East. Bennett Greenspan, president of Family Tree DNA, a DNA testing service, has also contributed to the understanding of Y chromosome types. According to Greenspan, among the 90,000 Y chromosome samples in his database, The haplogroup K2, now considered part of haplogroup T, appeared in 2% of Ashkenazi Jews and 3% of Sephardic Jews. Greenspan suggested that it was challenging to determine whether the non-Jews with K2 were descendants of Jews or if their lineage originated from earlier migrations into Europe. His sense leaned towards separate migrations from the Middle East, indicating complex historical movements of populations. In a broader analysis of YSTR loci of the University of Porto, Portugal, identified 41 different haplotypes spread across 10 haplogroups. The most frequent haplogroups among these were R1b1a, M269, J12f2.1, and T-M70, which together accounted for 80.7% of the total genetic samples analyzed. The high frequency of lineages like J12f2.1 and T-M70, typically predominant in Jewish populations, found in regions like Bragança, Portugal, was explained as a result of genetic drift in isolated, small-sized populations. However, the genetic diversity found within these haplogroups was surprisingly high, contrary to the expectations of genetic diversity loss due to inbreeding practices in the Portuguese crypto-Jewish communities. Haplogroup T, defined by the mutation M70, is particularly noteworthy due to its attributed origin in the Middle East. Despite its global rarity, its high frequencies in specific Jewish populations, such as Sephardic Levites, 23%, and Sephardic Israelis, 13%, underscore its importance in Jewish genetic studies. Further supporting the Middle Eastern origin of haplogroup T, ancient DNA, aDNA studies reveal its presence in significant archaeological sites like Abel Beth Makkah, Iron Age Levant, Tel Megiddo, Bronze Age, and Pekian Cave, Copper Age. These aDNA findings from the Holy Land establish a historical genetic continuity that threads through to modern Jewish diaspora communities, as evidenced by discoveries in medieval Ashkenazi sites, such as Erfurt in Germany and Norwich in England. Haplogroup T's distinctly higher frequencies among Jews from the Middle East, North Africa, Spain, Yemen, and Iraq, as well as among the Lemba, an African Jewish community, position it as a critical genetic marker for studying Jewish ancestry. It aligns with specific Jewish traditions and historical accounts, such as those of the Levites and other tribes like Naphtali and Dan. For example, the tribe of Naphtali traditionally inhabited northern Canaan, and their symbol, a swift gazelle, symbolizes their reputed speed, a trait potentially reflected in genetic markers prevalent among populations known for endurance running, such as those in the Horn of Africa. In summary, Michael Hammer's contributions significantly enhance our understanding of Jewish genetic history and population movements. His work integrates genetic data with historical context, offering a nuanced picture of how ancient lineages have survived and diversified over millennia. The tentative link between Thomas Jefferson's Y chromosome and Sephardic Jewish ancestry is a testament to the complex and interwoven tapestry of human genetic heritage, revealing how deeply interconnected our histories truly are.